Hi, our work is entitled Computing Control Laws for Gene Regulatory Networks within a Discrete Event Systems Approach. It was developed by me, Fábio Baldissera, and by José Curi. Unfortunately, we could not attend the conference and we apologize for that, but we prepared a video for you and we hope you enjoy it. Suppose we have a single cell, a human cell or a bacterium for instance, and we want to change its behavior. Our question is how to design synthetic genes so that when inserted into the cell, they enforce the behavior we want. This problem has relevant applications in biotechnology and medicine. It is a control problem, where the cell is the plant to be controlled, and the designed synthetic genes are the realization of our supervisor. The behavior of cells is mostly dictated by the states of each one of its genes. The state of a gene can be seen as a measure of the rate at which proteins are synthesized according to the instructions contained in this gene. Each gene, though, can also influence the state of other genes by means of the proteins they produce, giving rise to the so-called gene regulatory networks, the networks whose behavior we want to control. We then abstracted the behavior of gene networks by Boolean networks, where at a given point in time, each gene of the network can be either on or off, and where the interactions between them are modeled by Boolean functions. So for instance, if this gene is on, it represses this other gene. With these assumptions, it is possible to model gene networks with finite state automata, where each state of the automaton has the information of the individual states of each gene in the network, and where the events correspond to the activation or repression of a given gene. For this model, we devised algorithms based on supervisory control theory to find a control policy, which is a set of sentences like activate the gene that produces a given protein, let's say a Rabinow's regulator, if and only if the network is in a specific state X. These instructions can be implemented by a set of synthetic genes with the help of a branch of science called synthetic biology. We solved this problem in previous works, so now we start with our contribution for this conference, where we propose to extend our results to gene networks modeled by ordinary differential equations. Why? Because gene states are not simply off or on. There can be sometimes an almost continuous range of different rates at which a protein is synthesized from a gene and degraded in the cell. Therefore, many researchers model the behavior of gene networks by means of continuous formalisms instead of discrete event ones. In continuous models, we trace the concentration of proteins produced by genes. The rate of change of a given protein in the cell is equal to its synthesis rate minus the degradation rate. For instance, suppose that gene 2 activates gene 1. This could be modeled by the following equation, interpreted as the rate of change in the concentration of the protein produced by gene 1 is equal to the synthesis rate of this protein, a function of G2, given that gene 2 activates gene 1, minus the degradation rate, assumed here to be proportional to the concentration of the protein synthesized from gene 1. Note that the function for the synthesis rate has an S shape. When the concentration of the protein produced by gene 2 lies above a certain threshold, the synthesis rate of the protein produced by gene 1 increases a lot. Such functions and their generalizations for the case where multiple genes affect the state of a given gene are widely used to model the dynamics of gene networks. To obtain a discrete abstraction from the continuous model, we used a very simple procedure. Other procedures described in the literature could also be used. First, we partitioned the state space of the continuous model into regions of low and high concentration levels of proteins. We then selected a set of uniformly distributed points inside each partition element and mapped these points according to the ODEs that govern their dynamics until they crossed some boundary between partition elements. Crossing boundaries are the events that bring the system from one state to another. This procedure allows us to obtain a discrete event representation of the plant behavior, which can be simplified by eliminating those transitions that occur for just a few points. The simplified state transition diagram can be easily converted into a finite state automaton model of the gene regulatory network. With this finite automaton model in hand, we can compute the control policy exactly in the same way we did before. But now, instead of jumping to the realization of the control policy, we convert the logical control actions into continuous time equations, using the same reasoning adopted to model the behavior of gene networks, but now backwards. So, for example, if the control action is produce protein area C if and only if the cell is at a state where there is another protein, say Ci, then we can think of a synthetic gene GS that produces area C and whose dynamics is given by this equation, interpreted in the same way we did during the modeling step. But why do we have to convert the logical control directives into their continuous counterpart? Because kinetic parameters are important guidelines for the construction of synthetic genes. Logical control actions are important, but not sufficient for the construction of genes. Kinetic parameters of synthetic genes are related to important biological measures, such as synthesis and degradation rates of proteins, protein-gene binding affinities, stability of the intermediate molecules produced by genes called messenger RNAs, and interaction strength between a gene and the gene reading machinery in the cell, and so on. These are design parameters that, once defined, allow us to go to the wet lab to construct the synthetic genes. Our approach was tested on a gene network with 3 genes and a fixed set of kinetic parameters. We successfully changed the attractors of this network and could also enforce different trajectories to go from one initial state to a desired final state. The next steps in our work are assess other approaches available in the literature to obtain better discrete event abstractions, refine our approach with respect to the choice of parameters, threshold for low and high levels of proteins, kinetic parameters for the realization of the supervisor, and so on, and test the approach in vitro. For that, we have an ongoing collaboration with the biotechnology department at our university. If you have any questions, please feel free to contact us. Thank you.